things happening and key transcriptional events in particular that take place in pre-implantation development, especially that first lineage decision and the third lineage decision that have to do with pluripotent stem cells. Okay, because remember, and we'll go over this, embryonic stem cells are made from the ICM of embryos. Okay. Now we'll see, and you probably heard of these, there are also induced pluripotent stem cells that have all the characteristics and many of the characteristics of embryonic stem cells, but the induced pluripotent stem cells are fascinating because they're actually derived from somatic cells and reprogrammed. That's the part of the pluripotent stem cell lecture that I'm going to put off until next week. Okay. But anyway, there's also, so these cells, I've shaded them, but everybody should remember what an ICM is, right, versus the trophectoderm. These cells, though, are, functionally and in many respects molecularly, cellularly equivalent to the pluripotent state of stem cells. But what makes them pluripotent is that they can differentiate into all of the three primary germ cell lineages, and in many cases, the lineages that are developed from those three distinct ones. Those three distinct primary germ cells, the three primary, anyway, happens during gastrulation. And I think I showed this slide the other day. That is a disc. It looks like a shell, and there's a little groove forming there. That's what you all look like. This is essentially considered, it's called a bilaminar disc. There's essentially just two layers of cells. What that two layers of cells is going to do is convert into three layers. Okay, and a few other things. So just to kind of orient you to where we are, so here's implantation. We saw that video the other day. Now this is showing implantation of the embryo on the left side of the uterus. So the interior of the uterus is here. So the blastocele cavity would be on the interior, and this is invading in. Now the next slide, now what I've done, just to orient you, this is an electron microscopy. I've covered up the actual image on the posted slide. But now this is looking at, this is that same structure from the previous slide. I've just rotated it around so that it increases sound. So it's pretty much like this. So to the bottom would be the interior of the uterus. To the top is where the invasion is going. See these two blue? The blue and the yellow are the embryonic disc, the embryonic cells. Everything else is trophectoderm, the individual cells are trophoblasts, and these things are starting to differentiate. So here, you can't really make it out too well, but if you look real close, you can see just the two layers of cells. Now, an expert would be able to tell just from the morphology what they are. What then happens now, okay, so this is looking at kind of a side view. If we look down on that embryo, this is what you'll see. Okay. And this is that same figure from the couple slides ago. This is a blow up. This is the primitive streak. Primitive streak. What we're going to see, gastrulation, is cells moving from this top layer into the interior, but then some really fascinating things take place. So here again, this is another view of it. Here's the bilaminar disc, embryonic disc. This is not from the side view like we saw the EM. This is looking kind of laterally and to the top. There's the primitive streak. Don't worry too much about the difference between the node and the primitive streak and these other things. But this is where those cells are going to be going through. Now there's this video, I forgot how long it is now, but I think it's about 14 minutes. It goes much further. After about the first 2.5 minutes, it goes much further into what happens after gastrulation. You can look at it if you want. It's really cool what happens. I want you to focus on, and what we're going to look at today is just gastrulation. Okay. And remember, I want you to learn about what's happening in the embryo, because this is what's being studied intensely with pluripotent stem cells, how to reproduce this. And this is exactly what they do. So here's the embryo. Not a whole lot is happening at the level of the embryo here. This is just after implantation. Now it's embedded in, and you're getting all this differentiation about the extra embryonic cells as well as the maternal uterine cells. There's the bilaminar disc. Don't worry about this little stalk that's forming. Now start paying attention. Same color coding, apiblast and hypoblast. Okay. This is before the second lineage decision. This is only at the, I'm sorry, this is after the second lineage decision, sorry. Okay. Now, don't worry about this either. It's just gonna keep, the stalk, this is gonna eventually form into the placenta, don't worry. No, that's not, I forgot what that is, never mind. Yeah, maybe it is. That part of embryonic development, I don't know too much. Okay, gastrulation. This is a very important statement. Lewis Walford, very well-known and famous developmental biologist. I bet you didn't know that. Okay, now pay attention to those terms, dorsal and ventral, back and stomach. Okay. Now, cranial and caudal, head, tail, we don't have a tail. But, okay, as soon as this primitive streak forms, and it starts from the caudal end and starts moving to the other end, the cranial end, that immediately sets up left-right polarity, as well as the caudal cranial. And remember, we already had the dorsal ventral set up. So all the major body axes are set up. There you saw the cells migrating. Now we've turned, so we're looking at the cells migrating. But look at this. There's a transformation. That first wave of cells causes a transformation of that primitive endoderm to the definitive endoderm. Now we get another wave of migration, but these cells, these orange cells, are getting the signal from these, the definitive endoderm, as well as signals from the ectoderm to tell them they're in between those two other distinct types of cells. I'm a red cell, I need to stay here, I need to form what's called mesoderm. Okay, now this is, oh, there's just some really cool things going on here. Let me see if I can back up a little bit. Okay, so everybody see where now we've just turned this at the cross section. Now the blue cells are going to start migrating through. But they're going to, the first wave of migration, they're going to just, they kind of take, some of them take the place, some of them, once they start replacing the primitive endoderm, they cause what's called an epithelial to mesenchymal transformation. Don't worry too much about it, EMT, but it is an important developmental process, normal developmental process. It's another thing about embryonic development that shares some aspects with metastasis, with cancer cells. Okay. So the first wave of migration causes the transformation of the primitive endoderm into the definitive endoderm. Now that second wave forms that third layer. So now we've got ectoderm, mesoderm, and endoderm. Those are the cells, those are the primary germ layers. And those terms you need to know. This is what we want, well, we want to understand because in the embryo, because this is the most important event in your life, but also because this is what we want pluripotent stem cells to be able to do. Pluripotent stem cells are undifferentiated, like they would be in the ICM. But, and you can keep them that way, but what you want to do and what the whole field wants to do for both basic research and direct clinical applications is to be able to control the conditions in the media or with genes, adding or removing, knocking down genes, preferably with media, sometimes adding growth factors, sometimes adding chemicals to the media that will direct those pluripotent stem cells into either ectoderm or mesoderm or endoderm. Because once that first commitment is made, Then the next step in that differentiation with pluripotent stem cells would be to control, direct. Let's say you directed your pluripotent stem cells to differentiate into ectoderm. What you then want to do, and is being actively studied, is then take these ectodermal cells and differentiate them into, as we'll see, components of the central nervous system, components of the peripheral nervous system, and skin. Same thing with mesoderm, same thing with endoderm. Now, the things that are taking place both at the molecular level and the cellular level in pluripotent stem cells don't always exactly reproduce what's going on in the actual embryo. Okay? But studying one does give good information to test in the other. Okay, so going back and forth between them is really important. Okay, so this is just giving you that drawing again, same kind of color coding. Remember, endoderm, mesoderm. Oh, I should have got endoderm there. But remember, it's converted from the primitive endoderm hypoblasm to the definitive endoderm. And there's that statement again. Okay, now here's another drawing. Here's our 3 germ layers. Now let's see what they develop into. I do want you to know these. That's what I said. Epidermis, the skin, and the nervous system for ectoderm. Outside, ecto. Mesoderm, middle. Connective tissue. Now, so if you don't know, connective tissue, like if you look under the skin cells, the epidermis of the skin, there would be connective tissue, mostly made by fibroblasts and things like that. Think of that as kind of being the structural supporting cells, tissue, for the functioning cells that are above that. Same thing with any in your gut, in your lungs, anywhere in your body except for blood cells, where you've got a specialized type of cell being maintained by a structure underneath it. That structure is always mostly going to be connective tissue. That will be mesodermal derived. What there could be would be endoderm derived cells on top of it. The endoderm derived cells would be the specialized cells. Okay. Now the other thing too, this is kind of interesting, that mesoderm, in addition to making the connective tissue, also makes the blood cells. Red blood cells, white blood cells. Endoderm, essentially think epithelial cells, like in the gut, the lining of the gut. Okay, remember the polarized, when we saw in the 8-cell embryo, the polarization of those cells in the apical versus basolateral domain? Epithelial cells are polarized. They're in a single layer. They line the gut, lungs, and so far. We're missing a cell type though, germ cells. They're special. Remember, they're not part of it. They're the ultimate pluripotent stem cell. They're the only cells that contribute to the next generation. They're the only cells that once they're inherited by the offspring, they're the only cells also that are pluripotent. Right? So there's actually a lot of qualities, characteristics that are shared between germ cells and pluripotent stem cells. Okay? But that is a separate lineage. And if you're wondering, people are starting to discover the conditions where you can make eggs from induced pluripotent stem cells. They're not quite there yet to functioning, so it's kind of fascinating. Okay, do you want to learn this? No. But there's our embryonic endoderm. So here's, I put squares around the red rectangles around embryonic endoderm, mesoderm, ectoderm. But pick out your favorite cell type or tissue, the one that you want, you're most interested in. See where it's lineage. A lot of these, and the cells that are the tissues that are formed either in the fetus or postnatally are actually a combination of cell types derived from 2 different primary germ layers. Yes?
Do we consider endoderm, mesoderm, and ectoderm to be the first tissues, or are they not tissues at that point?
Well,
tissues are derived from them?
Yeah. I mean, you can call any group of cells. I mean, if it's, I don't know what the exact definition of a tissue is,
Okay.
but you could call the bilaminar embryonic disc the different, those could have different tissues, but let's, I guess more in terms of what they're doing, let's make the distinction. Let's not think of ectoderm, endoderm, and mesoderm as being tissues.
Okay.
Let's just, for our purposes, let's think of tissues as being, you know, either the organ that's in the process of developing or the fully functioning adult postnatal organ.
Okay.
And there can be different tissues within that organ. Yeah. Okay, this just kind of explains, if you wanted to watch the rest of that video, this ties in with what happens after gastrulation. And you can just take a quick look at these scanning EM of human embryos. Aren't they beautiful? I have to tell you, I think I might have said this, the research I did for 20 years was in mouse genetics, and a lot of it was in embryonic development. A lot of what I worked on with mouse embryos was later stage development, but then at one point I started working on the stage that's equivalent to this. And the first time I did these dissections under a dissecting microscope, anybody ever seen? Anyway, they're not like a microscope for looking at histological sections. But the first time I saw these embryos myself, I'd seen hundreds of pictures of them, but the first time I saw them myself, I almost fell off the chair because they were just so beautiful. I mean, beautiful from an artistic point of view, but obviously my artistic view is biased towards science. I'm not looking at that. But to think that this, and think of what happens before and after this, or this, that this developed from a one-cell zygote, but this also going forward is developed into us. There's the head fold. That's what's going to become the head. That we had folds in our head. Anyway. And this is just some more. Okay, next lecture. What is it? Yes, pluripotent stem cells. So what I'm planning to do is today, I hope to get through sections 1 and 2, because this is going to lay the foundation for you to understand the research papers for next week. This I'll come back to, ideally on Tuesday, or later in the semester. Okay, but just know that there are lots of connections between the embryonic stem cells that we'll be talking about this week and next week, and in these pluripotent stem cells. Okay, so there's some terms, you may already know some of these. You may have already learned pluripotent versus totipotent versus multipotent. Pluripotent is what we're focusing on with respect to pluripotent stem cells. Note that there is a difference here. Totipotent means all the tissues, cell types of the embryo, including the extraembryonic, what makes the placenta and other extraembryonic membranes. Totipotent cells, and germ cells too. Pluripotent can't do the extraembryonic tissues, can do all of the embryo. They're working on seeing if these pluripotent stem cells can be converted truly to germ cells, but that's kind of difficult because ultimately, even if you thought you derived germ cells from the pluripotent stem cells, you'd want to functionally test them and find out, well, if you think you've developed an oocyte from a pluripotent stem cell, you'd want to find out if that oocyte could be fertilized and then develop an embryo to term. Obviously, there's ethical questions about limitations for doing that with humans. You could test it in mice. You don't have to know this term, you might be interested in it, and then just know these other terms too. Now more terms, and I took these directly out of the textbook, so I think they're reliable. Lineage and commitment, they're very much related to each other. A cell type goes through a commitment to become a cell lineage. Basically think of it as the specialization, the steps that are taken, oftentimes epigenetically and at the transcriptional level, that program the cell on a defined differentiation pathway. Differentiation, specialization. Those pluripotent stem cells, the ICM, they are not differentiated. They're not, and differentiation means control of gene expression, so that only the genes that are needed for specialized function of that particular differentiated cell type are being expressed. Progenitor cells. Now, there are blood progenitor cells, there are multipotent hematopoietic stem cells that are the progenitors of all of the other blood cells, erythroid cells, white cells, and so on. Okay, so those would be the progenitors. They can divide a little bit, not to the extent, that you would see in a truly pluripotent stem cell, and they do have some specialization, relatively undifferentiated. Now, these two terms, I think I've been referring to them enough that you should know. And remember, these are both pluripotent. Now, what we'll see when we talk about iPSCs, there is what's called naive and committed pluripotency. We'll see a difference between later. Okay. Yes?
From a lab perspective, what are the benefits of using progenitor cells as opposed to like just regular stem cells or like embryonic cells?
Yeah, I guess it would depend on what you're doing. If you wanted to only be working with a specific lineage from a particular type of progenitor cell, there might be some advantages to doing that. If you wanted to have the capability to look at different lineages, maybe and compare results that you get with the two, or something like that, then you might want to work with embryonic stem cells. And the other thing is practical consideration. Embryonic stem cells are a little more tricky to work with, I think. You can learn them, and you have the techniques now are real, and reagents you need are real common now, not in the mid-90s, but now they are. So it kind of depends, but that is a good question. Yes.
Are there significant enough mutations to change the commitment of a cell from one type to another?
That's a good question. So are you talking about if you have, let's say you have, certain type of progenitor cells and you think they're supposed to be only going on a certain commitment, but you think there might have been a mutation that happened that's altering that?
Mm-hmm.
Yeah, that's something that you might have to consider. So you would all, with any kind of experiment, especially when you're using cells, you're going to be monitoring them. If you're good, scientists should be careful, and being careful means you're a good scientist. Okay, you would be being very careful about the behavior, morphologically, molecular characterization of your cell types of your cells. And if you ever saw something happening, even if it was just a small group of cells that seemed to be behaving differently, you didn't do anything to cause that, you would pay attention. Okay, now a lot of these, so embryonic stem cells, as well as, it just depends on what type of progenitor cells are differentiated. A lot of them can be frozen and stored, and then rethawed when you need them. So if you ever thought some kind of mutation had happened, you could just throw those out and go back to your frozen stock. But that's a good question that I'll come back to when we talk about embryonic stem or any kind of pluripotent stem cell. You're probably talking about mutations like change in DNA sequence. And you think of something else that could happen at the level of the DNA that might impact the phenotype of the cells that we talked about that can happen both in mitosis and in meiosis? That reminds me of them both. Can you think of something that can happen in cells? These are going through mitosis.
It's like non-distinct.
Yeah, exactly. So oftentimes, especially with embryonic stem cells, people regularly do karyotype of them just to make sure they haven't lost or gained a chromosome, because that can alter behavior. Is that what you were going to say? Yeah. The other thing, and I'll mention this when we talk about embryonic stem cells more, and you'll see it in the paper that we'll be discussing, I think both papers, they always have to go there, even if it's not in the the main part of the publication, the research paper, every paper has supplemental information, okay? And it'll refer to supplemental figure 1, supplemental figure 2. And I'll tell you this later, but for most papers that we're gonna be assigning, you won't need to look at any of the supplemental information. If there's something particularly important, I'll attach that to the PDF of the paper. The reason I'm talking about it now and want to mention it with embryonic stem cells and iPSC cells, once they've derived these, they have every science, before every publication, and you would do this just routinely, you always will be establishing and testing whether these cells truly are pluripotent and whether they have, and there's several measures of that, and whether they have a normal karyotype. It used to be that, like in the paper that we're talking about, the 2006 paper, see, they've got that in the stuff. They refer to it, they refer to it, they didn't actually publish any papers. It wouldn't have been uncommon in the mid-2000s to have that published. It's much more common now since it's more common techniques for that to be in the supplemental information, but it's got to be done because you got to know that they're pluripotent. Okay, one more thing, or two more things since it came up. These embryonic stem cells, remember they're derived from embryos, obviously, whether it's mouse or human. Can you think about anything that, let's say, you got one batch of embryos, let's just say human, because we're talking about human genetics. Let's say you got embryos donated from an IVF clinic, and you made two embryonic stem cell lines, one from each of two different embryos. So embryo 1, ESC line 1, embryo 2, ESC line 2. Can you think of any differences? You're kind of nodding your head like you know where I'm going. Yeah. Two lines, but remember each line was made from a different embryo. Yes. They're not going to be genetically identical. Could that be affecting any of the properties, like especially if you start looking at differentiation? Yeah. So that's one of the big concerns. Okay. And there's different ways of handling that. The other thing is anytime you put cells in culture, it's a selection for cells that can grow under those conditions. You know what selection is, like putting a plasmid with antibiotic resistance gene into bacterial cells that don't have that resistance, and you're selecting only for the cells that grow. That exact same kind of selection is taking place with these cells. And there's actually been studies, getting back to the non-disjunction point, there's actually been studies that have shown have intentionally looked for selection during in vitro culture conditions or changes in growth behavior that can be related to selection for a chromosome rearrangement or a particular chromosome trisomy or monosomy or anything like that. So yeah, all really important things to think about. Just from the historical, so mouse ESC cells were established ages ago, 1981. And these mouse embryonic stem cells were first used for making knockout mice. That's what they're most famous for. It was only until 1998 that the first human ESCs. And this paper raised a lot of controversy. In fact, I've even read some of the editors of that Science. The Science had an opinion piece about how they had discussed whether they should publish it, and all because of the ethical issues of the fact that these cells came from human embryos. Okay. Yes.
But clearly, they had to get past an ethics board in the first place so they could perform the study.
Yeah.
So at that point, I don't understand I understand how they're planning on publishing it, but the research is done. The ethics are over. The data's there. Why? I don't understand
Like why hold it
You have a
back?
scientific perspective on it. As a scientist, you know that you're going to get.
Yeah, the information's already been figured out. Why not? Like, I don't
But
understand
people
how it's
will
regulated.
object to the use of human embryos for anything. Some people, and I don't wanna get into whether, that's not a right or wrong issue, it's, well, I have my opinion, but, you know, it's my opinion, and not everybody else is entitled to their own opinion, right? And my opinion is informed as a scientist as much as possible. But keep in mind that it was, I should have looked up the dates, maybe somebody knows, but it wasn't too long after that. So I think in this paper, again, you don't have to, I'll show you a couple figures from it, but I think it was either Jamie Tons in the same lab, they generated 4 human ESC cell lines from 4 different embryos. And it was at some point shortly after that the government decided no more research on involving human embryos. So there was a ban for any scientist using federal funding like NIH grant money or NSF grant money, which would be just about anybody doing this kind of research. It was banned being able to develop any more ESC cell lines. They could still do research on the first 4 lines, but that was it. For federal funding, private institutions, research institutions, actually quite a few of them were formed, they weren't subject to the government ban. So a lot of scientists working in human embryonic stem cells either were recruited to work at these private institutions or got money for the research from them. Now that's no longer the case. And in large part, I think, because of the fact that we're learning so much from these, so they're very powerful. Also, I think people's opinions, it's just my opinion, my impression, people's opinions change a lot when scientists start making induced pluripotent stem cells. Those do not come from embryos. So maybe kind of the ethical questions got a little bit relaxed. My timer went off for a break. Anyway, let's take a break. This is happening. And they're irradiated fibroblast feeders, so oftentimes these are made from mouse embryos. What is really important, there's 2 things. One, the reason they're irradiated is so that they don't overgrow the cells, overgrow the dish, right? But what they're doing, so they're not going to be divided. There's still a lot of living cells, and they wouldn't be able to live forever, but they'll be able to live a week or so. Just basically, not dividing, but still expressing things that are needed on the cell surface of the feeder cells, as well as expressed into the media that is absolutely essential for the establishment of the embryonic stem cells and for maintaining their pluripotency. What we'll talk about in a little bit, if you take embryonic stem cells, whether they're human or mouse, away from the feeders and put them, let's say you can literally just kind of scrape off these cells, transfer them to another dish that doesn't have any feeders, those will immediately start differentiating. Now, what people didn't know back in the day, in 2006, is exactly what was being produced by those feeders that maintained the pluripotency of the stem cells, but something was, because they knew if you take the feeders away, you get differentiation. Nowadays, they've got more defined culture conditions, especially with small molecules, where they don't necessarily need the feeders. But you still have to have some kind of media conditions that keep those cells pluripotent. Again, this is just showing you another figure of the mice. Now, just of human in particular. So just another view, blastocyst, iceum. Now, so this is a magnification of actual human ESL colony. These flattened cells, you can probably see them better on your own laptop. These flattened cells, those are the feeders. I think for human ESCs, they're still using mouse feeders, but I'm not certain about that. But it's in the but it's right. Oh, and of course, we've already kind of talked about the ethical and political issues of human ESCs. Okay, so I use the term pluripotency a lot, but kind of implicit, if you're saying a cell type is pluripotent, you're implying that they also have this other characteristic, which is self-renewal. Yes?
Can you go back to the slide with the cell cultures? So really quick, what's the difference between ICM and the established embryonic stem?
Okay, so the ICM is really literally what's been, okay, so you can dissect this from the embryo. And then you would replace those. This I would still consider to be embryonic cells. If you didn't put them on feeders or the more recently developed culture conditions, these would not survive. You attach them, you put them in this dish with the feeders or under the other culture conditions, and then they become an established cell line. So they're not automatically, it's not an automatic process. It has to take place under very defined conditions. Once you've got these cells growing, then you could consider it to be a defined cell line.
So like just to make, because I think I don't really quite understand yet. At the end, it's more differentiated than how they originally were.
No.
It's just like they're just on feeders. And
They're the purple, well now they're calling these purple because they're not quite established, but once you know you can continue to maintain them, and by maintaining them, you let them grow to a certain size in one dish. Once they reach a certain size, then you transfer them to a new dish, so those will grow some more, then you transfer them again, and then once you can do that multiple times, you know you've got an established line, and that's what these would be called. Now these are not individual from different embryos. These would be from, there's a few cells, individual cells, but they can all grow. Okay, self-renewal, they divide, but they don't, neither of them becomes differentiated.
Could you go back to the so in vitro, would the ICM just be considered the embryonic stem cells because there's no feeder cell?
Those are pluripotent, but they've got all the goodies that are coming from the blastocoel cavity, it's before implantation, all the goodies that are coming from the mom after implantation, right? Yeah. Not necessarily the same things that are here, but some of the same things are happening. Okay. Self-renewal, they divide, but both daughter cells stay undifferentiated and pluripotency. You've got to establish that they can form all three primary germ layers. And don't worry about this, but just if you're wondering, this is a higher magnification of those pluripotent. If you see anything in a paper that says pluripotent, that means undifferentiated and capable of self-renewal. If you took those ES cells away from the feeders, see the difference in morphology, and again, you want to compare and look at this on your monitor. These are flattened cells, they're differentiating. These are really tightly compacted, they're undifferentiated. An expert can easily tell the difference between these cells. Now, what are the two assays that are commonly used to establish pluripotency? One of them is in vitro differentiation, basically changing the culture conditions. And the new culture conditions don't promote pluripotency, but they promote differentiation. But it has to be for all three lineages. Germ, teratoma is the second. It requires mice, nude mice, immunocompromised mice, because what they do is take these cells that they think are pluripotent and inject them subcutaneously, just under the skin, in these immunocompromised mice. So the mice aren't having any immune rejection of the cells. But the coolest, they form teratomas. Teratomas are the coolest type of cancer that you could ever imagine. Okay. But they form these tumors, and they do occur in humans. They're very rare. But you look at these tumors grow, and they've got characteristics of any cancer cell, right? They can be malignant, they can be local invasive, they've got all their growth properties. But unlike any of the other types of cancer cells, these will form structures in specialized cells that are derived from all three primary germ layers. So, endoderm, Where'd they go? Endoderm, endoderm, there, the gut-like structures, cartilage, bone. I've even heard and seen pictures of teratomas in humans that have teeth, literally teeth in them. Yeah, they're
And
very.
hair.
And hair. Any type of structure can develop in them. They're fascinating, but awful if you have one. Yes.
So as far as like the elimination of the cancer or the survival of the cancer, would it be easier for it to survive and harder to eliminate if it is a teratoma, like it is able to differentiate?
Most of the time they occur in testes and men. Sorry about that, guys. But, and there are things that can be done for that. But most men probably don't want that happening. But, yeah, anyway. But it's not, this, we're talking about it as an assay. You want it, so the tumors take a couple weeks to grow, probably 7 to 8, longer than that, 7. But then you just add a histologist, you have a pathologist, look at histological sections and identify alteration. This is the in vitro differentiation. Remember I said if you take those pluripotent stem cells away from the feeders and put them in a dish, it has to have media, liquid media, and the proper media, like to support them. And there's no feeders there, they'll just spontaneously start developing, but they'll develop into a little ball. And the little balls of cells will literally kind of be floating around in the media. What they will do is spontaneously start differentiating. Look at this, that's like an endoderm. And, you know, in some ways an expert could tell this morphologically. And further on, you start getting more things happening. But both with respect to morphology, you might get a good idea, but you would always establish it using markers. So there are proteins, genes that are expressed at well-known, very established, universally accepted as expressed only in this particular cell type. Okay, so that's what these are. This is fluorescence immunostaining. This is a marker for ectoderm. Don't worry about what it is. Marker for mesoderm. Marker for endoderm. Okay. You can also do RT-PCR. Does everybody know what RT-PCR is? Reverse transcriptase PCR. So this is making mRNA, purifying mRNA, and making the complementary cDNA copy, and then PCR amplifying that. Yes.
I had a question about the last slide. How do the cells know, like, I guess, what lineage to differentiate it to without growth factors?
That's the $1 million question. And so I think probably in some conditions they know. But remember how in, let's say think about gastrulation. Even that primitive streak formation, that's going to take place only because there's essentially signals that are coming from one end of the embryo closest to the extraembryonic membranes. And so that's what's going to start those cells to specialize to form the primitive streak. Once those gastrulation starts, and those blue cells, remember that the animation, the blue cells are migrating, they're receiving signals from the primitive endoderm underneath that to make them be migrating only in the primitive streak. That transforms the primitive endoderm to definitive endoderm. Then that second migration starts, and remember I said when that animation was going that those cells migrating in that second wave, now they know they're between the ectoderm and endoderm, and they know to stay there and become mesoderm. It's because of growth factors. In some cases, there might be some cell-to-cell communication, contacts being made, probably not, but I'm not ruling that out. That's what's being reproduced here. And then, so we don't have a primitive streak. We don't have the uterus there. We don't have the same organization of the primitive streak and that bilaminar embryonic disc, right? But these conditions are in some way reproducing that. Some aspects of this is known, some isn't. This is not the way you would want to differentiate cells. If you wanted to study, the molecular mechanisms or cellular mechanisms of what makes cardiac cells different from neurons. And you wouldn't use this you wouldn't use this differentiation. This is just kind of an assay, quick, easy, you don't have to do anything special. But you do get the 3 primary germ layers if the cells are truly pluripotent. So again, these are just markers for specific, well-established markers. Okay. Now, this is what very active area of research since probably slightly before you were born, everybody around 20. Yeah, kind of some shit that's been going on that long, but yeah. This is what we want to know. I should correct the statement. Yeah, it is. Maintenance of pluripotency. What's keeping these guys not differentiating, but able to self-renew? And what do we need to do to convert these to ectoderm or mesoderm or endoderm? What are the transcription factors? What's happening at the level of gene expression? What's happening at the level of chromatin structure that's affecting gene expression? What's happening at the level of signaling molecules, growth factors, and receptors? All of that. That's what people are trying to study, both from the basic science aspect of it. It's fascinating. It has fundamentally, we talk about these to study early embryos. Same thing to study pluripotent stem cells. Not only for the basic research, but for potential and really important clinical applications. Okay. Now, it's pretty clear. I mean, you could draw a similar kind of thing with any kind of biological process. There's all things that are related into a network. And there's this whole another field of systems biology, basically networks, just studying how these networks are formed. But signal transduction, just all the things that, I didn't even mention microRNAs. Non-coding regulatory RNAs. All these feed together. Now, I've got several slides that refer to a review article written by the 2 main authors, the 2 senior authors of the Leopold paper that we'll talk about. But pay attention to the dates. This review article was published in 2008. So it includes and summarizes some of the findings that were in the 2006 paper and other papers published around the same time. So some of what's in here is reviewing what was learned from the paper we're going to look at in detail. So this actually gives you a little idea of what their big picture of what their results meant. So in 2008, they made this model where there's these 3 transcription regulators are controlling whether some genes are actively being transcribed or some genes are not being transcribed. So here's my question. Where do we hear of OCT4, SOX2, and NOP4? Do those sound familiar to you? They should. Why are you're nodding your head?
It's one of the
The embryo, the pre-implantation maintaining pluripotency, so it's not too surprising. I mean, a lot of the studies focused on these as being good candidates, and it's very well established. These are considered to be the core transcription factors. But what's really interesting about them is that they're both positive and negative regulators. Okay, so depending on what other co-activators are present or co-repressors are present will determine whether they activate or repress certain genes. Okay, anybody know what POL2? What is that? And what does this arrow mean? And any particular polymerase, like DNA polymerase? RNA polymerase 3? RNA polymerase 2, all 2. Now, you might not necessarily know this. Maybe you learned it in the biochemistry. Anybody? Okay, so we got this little tail hanging off of POL2. Anybody know what these little stars? You can barely see them. And what's happened between POL2, right at this arrow, and POL2 that seems to be moving? You think this would be the 5' end of the chain, and this is the 3' end of the chain. This arrow represents the promoter. Okay, so this would be the 5' end. Polymerase is going to move that way. Is this polymerase exactly the same as the polymerase here, this actively transcribing? This is the initiation stage of transcription. This is the elongation stage. These little stars, you don't necessarily need to know this, but it might become, it's actually mentioned kind of in passing in the Leopold paper. When Pol-2 switches from the initiation stage to the elongation stage, it leaves out behind all those general transcription factors. But another thing that happens specifically to the polymerase is that it gets phosphorylated. And there are antibodies that recognize the phosphorylated form of Pol-2 versus the non-phosphorylated form of Pol-2. Where that's really important is what, as we'll see in the Leopold paper, they provided evidence that was confirmed from other papers that there are promoters that are basically poised to switch from the initiation stage to the elongation stage. And so it'd be very important to assay, be able to quantify elongating carbon atoms, actively transcribing versus poised. Now the other thing here, so look at these categories, okay? So all of these genes, and there's many more, this is just an example of them. These are all active. These are all being repressed. Even if you didn't know ectoderm, mesoderm, endoderm, if you want certain genes to stay active in pluripotent stem cells, but other categories of genes to be repressed in pluripotent stem cells and undifferentiated, what do you think would you expect? These to be the ones that could be important for maintaining that pluripotency, and the ones that are repressed could be the ones that are being repressed, because actively repressed, because once they're activated, perhaps they're responsible for driving differentiation. That's exactly what these categories are, and that was the first very striking, one of the major observations made by Leopold. The other observation, okay, this is a slide from previous, just to remind you. There's actually 3 things, and again, this isn't all strictly from the Leopold paper, but it's very well established by 2008 and confirmed since then. OCT4, SOX2, and NANOG actually have 3 major functions. They activate transcription of a set of genes. They repress transcription of another set of genes. There's very important functional differences between the repressed and the active genes. But in addition, they have autoregulatory loop. They maintain their own expression. And all 3 of them together are doing that. Then the thing that focuses on with the Leopold paper and the Collinson paper on Thursday is the role of these proteins with a funny name, polycomb complex proteins. They associate, and the first evidence for this, it was from the Leopold paper and another one published from the same lab at about the same time, that these polycomb group complexes, PCG complexes, associated with the repressed gene, not with the active gene. So polycomb function in pluripotent stem cells is really important. Not having anything to do with expression of the genes that are maintaining pluripotency, but if you don't have polycomb function, what do you think would happen if you didn't have, let's say you had pluripotent stem cells, and let's say you had a way of either knocking down polycomb or making genes that cause polycomb defective, what would happen to those embryonic stem cells? polycomb's role is to repress these genes that are important for differentiation. If you don't have polycomb function, what happens? They start differentiating, yeah. You wouldn't necessarily be able to predict that they would go into one lineage or another, but they would certainly start differentiating. That would be that, and that's actually what we'll discuss in the comments in the call paper. Further analysis of what happens with polycomb in pluripotent stem cells. So I mentioned that the promoters are poised. That was the other surprising thing that the first evidence for came out of the Leopold paper, and again was followed up. Now this is a much more recent review, so a lot has gone into this. You don't have to necessarily know about MLL. It's actually it's a gene that was first identified from human leukemia cells. Initially didn't known to have any role in transcription, certainly. But you know what is this funny nomenclature? What is H3K4ME3? And this one is, it's about the same, but it's K27 instead of K4. What the heck are those? Which particular histone? 3. Histone 3, so is this in the core nucleosome or the linker histone? The core nucleosome is one of the 4. What's K4? Lysine 4. So the 4th amino acid from, is this at the N terminus or C terminus? N terminus, it would always be from the N, the numbering goes 1 to whatever. So the 4th amino acid at the N terminus of histone 3 is lysine. In this case, it's been trimethylated. So it could have been acetylated by a different enzyme. It could be monomethylated, bimethylated, dimethylated, or trimethylated. This specific one is trimethylated. This is trimethylation of histone 3 at lysine 27. Now it's very well established. You've heard the term histone code. These are the only 2 specific histone modifications that I want you to know, because these are 2 of the most well established. This is very diagnostic and indicative of active transcription, or the ability to be actively transcribed. This is repressive. K27 trimethylation is made by a polycomb group. That's the basis of the repressive effects of polycomb on transcription. This one is active, but look at this. Now they didn't. The Leopold paper doesn't have anything about MLL in there. I'm not sure it was even known at that time, but they did discover polycomb, RNA polymerase 2, and histone 3 K27 trimethylation. And the genes that are being actively repressed in pluripotent stem cells, and not on the genes specifically at the promoters, and not on the promoters of genes that are actively transcribed. But what was surprising is that they found that both these active, and later on they found both active and repressive marks found at these cells specifically that need to be activated for development. Because the idea behind this, and this actually made sense, right? Remember, those genes are not being transcribed, those developmentally important genes are not being transcribed in the pluripotent state, yet either in the embryo or in culture, those genes have to respond quickly to signals that are telling them to differentiate. So this would make sense to have those really crucially important genes be ready to go, right? Essentially, literally poised, ready to go. And that would just mean removing polychrome, removing this lysine 27 methylation, and the RNA call 2 is ready to go. It'll just go into the location phase. Okay. And this just tells you a little bit more. And this just tells you, yes, that this is more recent. Probably if you looked at even more recent than 8 years ago, you'd see even more things. But now we've got microRNAs. But it still follows the same thing. Now, if you ever see Tau5F1, that's the formal nomenclature for the gene that encodes OCT4. I know the protein is R4. I know it's confusing, but okay. Well, you know, I told you I would preview. Okay. Oh, yeah, I did get through that. I told you I'd preview the methods for the Leontal paper. How would you feel? I didn't get to it today, but I want you to really, if you don't